Thanks Organization Committee to give me this opportunity to share my research. I'm Xinyuan Chen, faculty at Biomedical and Pharmaceutical Science Department at the College of Pharmacy at the University of Rhode Island. University of Rhode Island is a state-owned public university. The main campus is located in Kingston Village, which is about 30 minutes away from the capital of Rhode Island. University of Rhode Island has about 15,000 undergraduate students, 2,000 graduate students, and 3,800 faculties and staffs. This is part of the Kingston campus. College of Pharmacy is in this elongated building. We are home to over 750 PharmD students, 140 biomedical and pharmaceutical science students, and 60 graduate students. Although URI is a relatively small state university, URI College of Pharmacy was ranked 8th in total federal research funding among 143 pharmacy colleges in 2019. My lab is mainly conducting drug and vaccine delivery researches. We use laser to generate tiny skin channels to facilitate transdermal drug and vaccine delivery. We are developing novel physical adjuvants to boost vaccination. We are also developing novel virus-like particle-based vaccine delivery platforms. Today, I will introduce a novel virus-like particle platform recently developed in my lab and its applications for vaccine development. Vaccine delivery platforms are used to efficiently present vaccine antigens to innate immune systems. First-generation vaccines, such as whole inactivated and live attenuated vaccines, don't need vaccine delivery platforms because they can induce potent immune responses. Second-generation vaccines, such as subunit vaccines, have relatively low immunogenicity and often require vaccine delivery platforms to be effective. Common vaccine delivery platforms include highly immunogenic proteins and virus-like particles. Flagellin and KLH are two representatives of soluble protein-based vaccine delivery platforms. KLH oligomerizes to form barrel-shaped structure and is highly immunogenic, and it's also a popular choice to prepare conjugate vaccines to induce high titers of antibody responses in research. Virus-like particles are made of self-assembled viral proteins, contain no genetic materials, and don't replicate in vivo. Virus-like particles can be divided into naked or enveloped. Naked virus-like particles only contain self-assembled viral proteins, while enveloped virus-like particles also contain host cell membrane components. Virus-like particles can be prepared from different viruses for exploration as vaccine delivery platforms. Other vaccine delivery platforms include liposomes and nanoemulsions, which are often used for co-delivery of the vaccine and adjuvants. Vaccine delivery platforms often act on antigen-presenting cells, such as dendritic cells, to improve antigen uptake and presentation, which is crucial for induction of antigen-specific T and B cell responses. Presentation of antigens on virus-like particle surface can increase antigen uptake, considering virus-like particles can be more efficiently taken up by dendritic cells than soluble proteins. Virus-like particles can also deliver antigens to MHC class I molecules, which is called cross-presentation. In contrast, soluble proteins are often presented on MHC class II molecules. MHC class I presentation is critical to stimulate cytotoxic T lymphocyte responses, which is crucial to get rid of intracellular pathogens and to treat cancer. Flagellin as a vaccine delivery platform can increase expression of co-stimulatory molecules and stimulate dendritic cell maturation. Dendritic cell maturation is crucial to stimulate potent antigen-specific T-cell responses. Most of the vaccine delivery platforms can increase antigen uptake and presentation on MHC II molecules, while some can induce cross-presentation of antigen and stimulate dendritic cell maturation. Since we work on high-density display of flagellin on HBC virus-like particle surface, we next focus on introducing flagellin and HBC virus-like particle-based vaccine delivery platforms. Flagellin is a major component of bacteria flagella filament and a potent toll-like receptor 5 agonist with potent vaccine adjuvant effects, which makes flagellin a highly immunogenic vaccine delivery platform. Flagellin contains 4 domains. D0 and D1 domains are highly conserved, while D2 and D3 domains are highly variable. D1 domain plays a crucial role in activation of toll-like receptor 5. And foreign antigens can replace D3 domain on insert to its N or C terminus without affecting its toll-like receptor 5 activation ability. Flagellin has been explored to develop vaccines against bacterial, viral, parasitic diseases, and cancer. Flagellin-based influenza M2E vaccine and hemagglutinin antigen vaccine have been tested in multiple clinical trials. Overall, these vaccines can induce high levels of antibody titer, yet all these vaccines have been found to induce a high risk of systemic adverse reactions. For example, they can dose-dependently increase serum C-reactive protein levels. C-reactive protein is an acute phase in an immune response marker. Flagellin-based vaccines were found to also increase serum interleukin-6 levels and induce flu-like symptoms in some patients. These adverse reactions were believed to be caused by an overt activation of toll-like receptor 5. Hepatitis B core protein self-assembles into virus-like particles with a diameter of 30 to 34 nanometer. There are about 100 spikes protruding from the HBC virus-like particle surface. The CE1 loop at the tip of the spikes provide an ideal site for insertion of foreign antigens and display on virus-like particle surface. However, only short antigenic epitopes and full-length proteins with closely juxtaposed N and C termini can be inserted without interference of HBC virus-like particle assembly. This limits the broad application of HBC virus-like particles as vaccine delivery platforms. To address the shortcomings of the flagellin and HBC virus-like particle platforms, we proposed to generate high-density flagellin displayed HBC virus-like particles. This approach is supported by the fact that N and C termini of flagellin are proximate to each other, which is less likely to impose significant steric hindrance to self-assembly of HBC virus-like particles. High-density display is also likely to embed D1 domain of flagellin in interior of virus-like particles, resulting in significantly reduced toll-like receptor 5 activation ability and leading to improved systemic safety. Highly variable D3 domain on the surface of virus-like particles can be replaced with foreign antigens with fewer restrictions on 3D structure, and thus the resulting virus-like particles may provide a more versatile delivery platform than HBC virus-like particles in vaccine development. We constructed a recombinant plasmid by insertion of flagellin gene into the CE1 loop encoding region of HBC, followed by E. coli expression, purification, and self-assembly. We found the flagellin HBC fusion protein can self-assemble into virus-like particles. The resulting FH virus-like particles have a size slightly bigger than HBC virus-like particles, as revealed by DLS and TEM analysis. Immunogold staining TEM confirmed the presence of flagellin on FH virus-like particle surface, and based on the immunogold staining TEM, FH virus-like particles are about 2 times bigger than HBC virus-like particles. DLS and TEM analysis probably didn't capture surface-displayed flagellin, thus leading to underestimate of FH virus-like particle size. We found a high-density display of flagellin on HBC virus-like particles led to significantly reduced Toll-like receptor 5 activation ability. This is likely due to the embedding of D1 domain in interior of virus-like particles. In contrast, soluble flagellin and a commercial flagellin showed a strong ability to activate Toll-like receptor 5. We further explored whether reduced Toll-like receptor 5 activation could lead to reduced adjuvant effects. In this study, we simply mixed FH virus-like particles with immunogen albumin, and we found FH virus-like particles similarly enhanced anti-albumin antibody titer to soluble flagellin in both prime and boost immunizations. This study indicated FH virus-like particles preserved its adjuvant effects despite the significantly reduced Toll-like receptor 5 activation ability. This result is in line with the literature that a strong activation of innate immunity is not required to achieve flagellin's adjuvant effects. Both flagellin and FH virus-like particle immunizations. Considering MID88 is a critical adapter mediating most Toll-like receptor, including Toll-like receptor 5 mediated pathways, these results are kind of expected. More directly, we also measured rectal temperature before and 24 hours after weekly injection of the 2 different materials at the same flagellin dose. We found that soluble flagellin could significantly increase rectal temperature, while FH virus-like particles could not do so. This study provides direct evidence to support FH virus-like particles have better systemic safety than soluble flagellin. Next, we explored whether improved safety may compromise the ability of FH virus-like particles to induce anti-flagellin antibodies. We explored this in wild-type and MID88 knockout mice to see how lack of MID88 affects anti-flagellin antibody production. After prime and booster immunization, at the same flagellin dose, we measured anti-flagellin total IgG as well as subtype IgG1 and IgG2C antibody titer. And we found FH virus-like particles could induce more than 2 times higher anti-flagellin antibody titer than soluble flagellin. Considering flagellin is highly immunogenic, this result is significant, although 2 times increase is not that impressive by just looking at the number. Interestingly, We found a soluble flagellin mainly induced IgG1 antibody titer, while FH virus-like particles mainly induced IgG2C antibody titer. These results indicated that soluble flagellin mainly induced Th2-biased antibody responses, while FH virus-like particles mainly induced Th1-biased antibody responses. This shift is likely due to the display of flagellin on virus-like particle surface. We further found MYD88 played a more critical role in flagellin-induced antibody production, considering we still observed a significant antibody levels after FH virus-like particle immunization in MYD88 knockout mice. Interestingly, when we analyzed anti-HBC antibody titer, we found FH virus-like particles only induced very low level of the anti-HBC antibody titer. This indicated that HBC is likely this indicating HBC was embedded in the core of FH virus-like particles and cannot be recognized by immune systems. Next, flagellin was used as a model to explore relative immunogenicity of FH virus-like particles to flagellin and HBC virus-like particles. Nicotine vaccine was proposed as a novel therapy against nicotine addiction. Nicotine vaccine is also called nicotine shots. Nicotine shots stimulate the body to generate anti-nicotine antibodies to bind and block nicotine entry into the brain. Thus, smoking and related highs can be disconnected. Nicotine vaccine concept has been proved in clinical trials. However, only 1/3 of the smokers can develop a relatively high anti-nicotine antibody titer to have a positive effect. Improving immunogenicity of nicotine vaccine remains the key for successful anti-nicotine immunotherapy. Considering nicotine vaccines are generated by conjugation of nicotine hepatine to vaccine delivery platforms, immunogenicity of vaccine delivery platforms is very important. The most advanced nicotine vaccine uses REPA as the vaccine delivery platform. REPA is a recombinant bacterial protein which has less immunogenicity as compared to KLH. In our study, we first compared relative immunogenicity of flagellin and HBC virus-like particles to KLH, and then compared relative immunogenicity of FH virus-like particles to KLH for nicotine vaccine development. We found that KLH conjugated nicotine vaccine could induce more potent anti-nicotine antibody production than flagellin and HBC virus-like particle conjugated nicotine vaccine. This indicates KLH was more immunogenic than flagellin and HBC virus-like particles for nicotine vaccine development. So how about FH virus-like particles? We found FH virus-like particle conjugated nicotine vaccine could induce the higher anti-nicotine antibody titer than KLH conjugated nicotine vaccine. After intravenous nicotine challenge, significantly lower brain nicotine levels were found in FH NIC group than that in KLH NIC group. Furthermore, we found the incorporation of CpG and alum adjuvant into FH NIC immunization could increase anti-nicotine antibody protection. This study indicated a potential synergy between flagellin and CpG or alum to boost vaccine-induced immune responses. Our study proved that FH virus-like particles were more immunogenic than KLH, and KLH was more immunogenic than flagellin and HBC virus-like particles for nicotine vaccine development. FH virus-like particles also hold a great promise for other conjugate or recombinant vaccine development. FH virus-like particles are highly immunogenic due to the incorporation of flagellin with potent adjuvant effects. FH virus-like particles have a good systemic safety due to its weak toll-like receptor 5 activation. FH virus-like particles also have a good versatility, considering its D3 domain can be replaced with vaccine antigens, with fewer restrictions on its 3D structure. So in summary, we prepared high-density flagellin-displayed HBC virus-like particles, which shows good safety, high immunogenicity, and versatility for vaccine development. This work is mainly conducted by my PhD student Yiwen Zhao, and this work was mainly supported by my startup fund and partly supported by my NIDA R00 fund. Equipment used in core facilities is supported by Rhode Island IMBRI and Corbry funds.